Right, so our next speaker is Medzhitov from Yale University. We'll be talking about post-defense strategy.
Thank you, Martin, for inviting me here. And I'm mostly here to support the cause and to learn. So my talk would be having this suspicion that everything I will have to say in the is extremely trivial and obvious, but all time. So it's really an amateur perspective on cost and meaning. This is a subject. Everything I know about it, I learned from reading papers written by good people in this room. So what I'm actually will focus on mostly today is on the question of how the costs of immunity or generally of trade-offs can be deconstrained. So the importance of trade-offs is well appreciated, and the ability to limit optimal functionalities in various contexts are also well understood. But one issue that is, I think, less well understood is how evolution develops mechanisms to deconstrain certain trade-offs, couple processes that constrain everything. So that's what I will talk about today, and I'm not going to really talk about specific data because it's not really my field, but I'll just use a few examples to illustrate the point. So to put it in perspective, there are three, very generally speaking, three strategies of host defense from infection: avoiding infection, resisting it by reducing the pathogen burden, and tolerating the presence of pathogens. And each of these strategies has their own types of costs listed here. In the case of resistance, that's where it's most understood. It's immunopathology as well as sickness behaviors that work at the expense of other types of more useful behaviors. And in the case of avoidance, it's also the process that it occurs at the expense of other adaptive behaviors like feeding, breeding, and so on. And in the case of tolerance, the main cost presumably that it occurs at the expense of optimal tissue functions. Probably the simplest example would be when column epithelium is exposed to mechanical damage or irritants, it can undergo metaplasia and become squamous epithelium. The absorptive function will be compromised, but the barrier function will be enhanced. So that's a trait. That's the cost of increasing tolerance to damage in this case. One can also look at specifically talking about costs of resistance. That there are different types of immune response in mammals, and mostly we'll use mammals as an example. And these are defined by the target of the response in the intended outcome. And they, of course, are they involve really different types of pathogens. So one class of pathogens are viruses and bacteria that infect cytosols of the host cells. The main target of the response in this case is the infected cell, and the intended purpose is to eliminate infected cells. And that can lead to certain types of immunopathology that in classical literature is called hypersensitivity step 2 and 3. The second class of pathogens would be extracellular bacteria and fungi, as well as bacteria that reside in vacuoles of infected cells, mostly macrophages. The intended target of the response here is the pathogen itself, unlike this case. And the intended consequence is to kill and eliminate the pathogen, and the type of hypersensitivity is type 4 here that can lead to pathology. And this is due to collateral damage mostly by things like reactive oxygen species and so on, or neutrophil proteases. And the third main class of infection is infection with multicellular parasites. The main target of the immune response here is not the pathogen itself for the most part, except for the larval stages. But in the case of adult worms, the main target of the response are the host, certain types of host tissues, and specifically it's mucosal epithelium. Endothelium is smooth muscles, and endothelium is proximal muscles, and the intended consequence of the goal of the response is not so much to kill the parasite. Killing adult worms is really problematic. So the main strategy of defense here is the expulsion of the parasite, and that can lead to characteristic pathology known as type 1 hypersensitivity or allergy, because it has to do with hyperactivation of these types of host tissues. Now, so the cost, because the cost of immunity can constrain optimal host defenses, the question is, can these trade-offs be deconstrained? And they presumably can be deconstrained, at least to some extent. And so what I'll discuss a couple of examples of this type of deconstraining of the trade-offs associated with cost. One has to do with specific strategies of regulation of the inflammatory immune response, and the other has to do with distribution of the costs and the associated hierarchy of the effector responses. So first about the regulatory strategies of the inflammatory response. I can use interchangeably inflammatory immune response here. In my example, I'll use the inflammatory response because it's simpler to explain the points, and you can imagine how it can be extended to the immune response as well. So inflammation in general is a reverse. It's an adaptive response to any adverse conditions. Classically, it's an infection, tissue injury, but depending on the type of inflammation, there are different types of less well understood conditions and triggers of the response, particularly the ones that are play a major role in many modern human diseases like obesity and diabetes. There, the triggers of the inflammatory response in that case are not well understood, and it's a chronic and low-grade inflammation associated with these conditions. It's very important in terms of its health implications, but it's relatively poorly understood. In all cases, this adaptive response to an adverse condition occurs at the cost of normal physiological processes, and for that reason, inflammatory responses are inducible in most immune responses. They're inducible because they have a cause. And that's what Rudolf Virchow called the fifth cardinal sign of inflammation, the loss of function. And first, I will give an example of regulatory strategy that has to do with distinction between what is regulated, the magnitude of the response, which is the main focus of research on regulation of immunity and inflammation. But there's another strategy is to control degree of responsiveness, and so I'll just give this quick example. Inflammatory response can be briefly summarized as follows, that it consists of four components. There are inducers of inflammation. Again, classically, this would be infection and tissue damage, but other types of less well understood inducers can be involved, such as loss of normal tissue functionality or homeostasis, also can lead to a chronic inflammatory response. There are sensors of the inducers, and sensors could be understood both as cells that are involved in sensing and specific receptors. Again, I'm using mammals as an example mostly, and the types of sensors that are involved are generally macrophages, but also mast cells and dendritic cells can also be. When they sense the inducers of the response, these sensors produce a variety of inflammatory mediators. In mammals, there are lots of inflammatory mediators, and it's an extremely complex system with, perhaps partially because we don't understand it well. It's basically, if you try to understand it, it's mostly a laundry list of different things and things that they can do. But these are mediators that are produced by sensors, and what they do, they affect target tissues, various target tissues. Target tissues have receptors for inflammatory mediators, and what these mediators do to target tissues, they alter their functional state. They alter their functional state in a way that is intended to optimize elimination of the inducers, whether it's an infection or tissue injury. Alteration of the functional state of the target tissue so occurs at the cost to the normal function of the target tissue. For example, effect on mucosal epithelium can be an increased production of mucins and novel cell hyperplasia, and that can affect, for example, absorptive function of the epithelium. Effect on epithelium can affect normal protein composition in the blood through effect on the liver. Effect on endothelium can have other consequences, obvious consequences, inflammation, and so on. So all these alterations in the functional state of the target tissues caused by inflammatory mediators happens at the cost, but the intended purpose is to eliminate the inducers. So there are 4 components of the inflammatory pathway, and each one of them potentially can be a subject of regulation. The first and most obvious way that the immune or inflammatory response, all these applies to immune response as well. The most obvious way it can control itself is by having a successful outcome of the response. That the response then leads to elimination of the inducers. For example, infection can be cleared, damaged tissue repaired, and so forth. And this type of control, it's interesting that this simple negative feedback control is all it takes to control certain types of biological pathways. For example, basic metabolic pathways are controlled in this manner, and that's all we need to control them, lipolysis and so forth. In those pathways, simply having a negative feedback usually by product of the pathway on some rate limiting enzyme is all you need. And the system will be extremely robust, not vulnerable to alterations, and there are no diseases other than genetic diseases that would be associated with dysregulation of these pathways. That's not the case for many other types of pathways, particularly for an inflammatory pathway. Simply eliminating the inducer is not enough. And we know that because if you remove any of the other types of regulators, that can lead to diseases associated with inflammatory tissue damage. The reason that simply eliminating the inducer is not enough in this case is because the effects of inflammatory mediators outlast their presence. And simply blocking the response, blocking the induction of these mediators is not sufficient to prevent immunopathology. And for that reason, inflammatory response is controlled at all other steps. So at the level of sensors, there's a regulation of the activation threshold of the response. And this is one of probably the most studied mechanisms of regulation of inflammation or immunity. This generally works through inducible expression of negative regulators of specific signaling pathways that are activated by inducers of inflammation. The second step of regulation is at the level of production of inflammatory mediators. The one I mentioned, various classes of cytokines, chemokines, and lipids, and so forth. And this is also relatively well appreciated and studied because many negative regulators of the inflammatory or immune response act at that level, like controlling production of inflammatory mediators. What is not well appreciated and studied is control at the level of responsiveness of target tissues to the inflammatory mediators. And there are 2 types of control at that level. One is